Hello, hi, a very happy good evening. So this is Krishnaveni and we've been talking about the story of life on earth, and that's biology. Yes, welcome to my session. So today we are in lecture 4 of molecular basis of inheritance. Today we're going to talk about the process of transcription. So how transcription happens in prokaryotes and how the process of transcription happens in eukaryotes, and what is the difference between the process in both these cases. So that is the agenda of today's class. So welcome to my class for all those who are watching my lecture. So thank you so much, and please do like, share and subscribe. Yes, Antonia B, a very happy good evening. So thank you so much for joining my lecture today. Yes, I'm good, thank you. How are you? Yes guys, so please don't forget to like, share and subscribe. Come on, make it quick. So then I will quickly start off with the process of transcription. So I guess by now replication process is pretty clear to everyone. So here we go to the process of transcription. So before we go to the process of transcription, so here I go. So this evening we are meeting once again at 6 p.m. talking about a new chapter, that is breathing and exchange of gas. So recently we have completed digestion and absorption. So today we are starting with a new topic, that is breathing and exchange of gases. So please join me for the same. Okay. So one more announcement. So once again, so we are starting a crash course for 45 days. So the class begins from 15th of July. So now we have an offer price going on. So the offer is 1,999. You get this package. So you have all the 3 subjects in this course. So you have P, C and B. So you have 3 subjects. So every day you have 3 lectures, and as well as every Saturday we'll have doubt classes, and Sunday you will have unit tests. So the offer is running right now because the actual price is 4,500. So now the price is 1,999 and I am handling biology. The course starts from 15th of July, so please join me for the same. I wish to see you there, but that's a bit a closer platform where we get to have one-on-one interaction. Okay, so make sure you join with me for the same. Alright, yes. So shall we go into our chapter, that is the process of transcription. So here we go. So first we're going to talk about the process. We spoke about the process of replication. So today, actually I started off with transcription. I don't know if you remember. So I gave you an introduction to the process of transcription. So today we'll go in detail about it. Yes, a very happy good evening, Darni. Yes, I'm good, thank you. How are you? So how was your weekend, everyone? So your weekend was good? Yes, my weekend was good. So thank you. So now we'll talk about the process of transcription. Before we talk about the actual process of transcription, we should know what is a transcription unit. So what comprises of a unit. Then we'll talk about transcription unit and the gene, how it is related, the types of RNA and the process of transcription. So the actual process of transcription is here. So today we'll try to finish off the process of transcription. Then I will start with the process of that is translation, which includes your genetic code, your mutation and genetic code. We'll talk about your tRNA, and finally the actual process of translation. Yes, Guha, hello, a very happy good evening. Thank you so much for joining my lecture. So please do like, share and subscribe. Alright, so here we go. So I hope all of you remember the process of replication. So once again, we have been talking about the process of central dogma of life. So in central dogma of life, the DNA replicates. The DNA faithfully produces copies of itself in the S phase inside the nucleus. Then the process of transcription begins, where the information on the DNA is copied onto the RNA. Right? Finally, the information on the RNA is decoded as proteins, and this is the process of translation. Here? Yes? Alright, so recap of replication, I hope you remember. So we'll only talk about the enzymes involved in the process of replication. So what are the enzymes involved in the process of replication? So as you tell me the enzymes, you will remember the process. So first we have the helicase, so which unbinds the DNA. Then you have topoisomerase, which relieves the stress on DNA. Right, so helicase, topoisomerase. Then you have primase, which produces primer, so which goes and binds to the template strand wherever there is ori site, that is origin of replication. Then number 4, you have the most important layer, that is DNA polymerase. So this is DNA dependent DNA polymerase, and this can catalyze the synthesis in the 5' to the 3' direction only. right? And it cannot initiate the process of polymerization. That is why we have the primer in place here. Here, yes? Okay, so this is the structure. So then you have one leading strand and one lagging strand. So in the lagging strand, you have Okazaki fragments. So these Okazaki fragments are joined together later by your DNA ligases. Clear? So this is in a nutshell the process of your DNA replication. So here I told you, your DNA polymerase also has a proofreading activity. So it has a proofreading activity in the 3' to 5' direction. Right? And the DNA polymerase, in case of your prokaryotes, it is of 3 types. DNA pol 1, 2 and 3, and pol 2 is used for the process of replication. And when you talk about your eukaryotes, you have 5 DNA polymerase, alpha, beta, gamma, delta, and then you have epsilon. So here epsilon is used for your DNA replication process. Your DNA replication is a highly energy expensive process, and your nucleotides act as substrates for the enzyme DNA polymerase, as well as they provide energy for the process of replication, right? And this process is very, very accurate, because if there is an error, it leads to the process or the consequence of mutation. Done? So this is a quick recap of replication. So now talking about the process of transcription. So I already began or explained what is transcription. So transcription is copying of the DNA sequence onto the mRNA, right? But now if I have 2 strands of DNA like this, but information from only one strand is copied onto the RNA. Why so? Though I have 2 strands of DNA, so both the strands are replicated during the process of replication, but information from one strand is only copied onto the RNA. So why so? So why so? So I give you the reason, right? So you have only information being copied on one strand from one strand like this. So this is the process of transcription. Why? So point number 1, so if a small piece of your DNA can produce 2 mRNA, if this information is copied onto this mRNA and this information to another RNA, so thus from a small chunk you will have 2 different proteins, right? So that is the main reason it is not happening so. Point number 2, these 2 sequences are complementary to each other. So if this sequence is copied onto an mRNA and this sequence is copied onto the mRNA, so in a nutshell you are producing double-stranded mRNA, because both the mRNAs will be complementary, right? Sound is not clear. Okay, just hold on, I'm coming. So now is it clear? Now is it clear? I hope it's clear. Okay, so let's continue. Yes, so one bit of DNA is only copied, that is because otherwise it leads to the formation of a double-stranded mRNA, which further inhibits the process of translation, right? Okay, so this is the reason why only one strand is copied. Okay, and since your DNA is also a nucleotide, your mRNA is also a nucleotide, the complementary base pairing drives this process. Clear? Is this much clear? So now if you have two strands and information from only one strand is getting copied, so now which strand is going to be used as a template? So that is the next question. Now which strand will be used as a template? So here we go. So for the process of transcription, you have an important enzyme, that is your RNA polymerase. So this RNA polymerase can only catalyze the synthesis again in the 5' to 3' direction. Thus the DNA strand which has 3' to 5' is used as a template strand. So this strand of DNA acts as a template strand for the process of transcription. Okay. So this template strand will catalyze the mRNA molecule, that is from 5' to 3'. So this is your template strand. This is your template strand. This is by nature is given a weird name. So this is called as a coding strand, or this is known as a sense strand. This is known as antisense strand. Sense, antisense strand. Okay. So this is your template strand. This is your non-template strand. Clear? Yes. So this is the first part. Now you have a next thing, what is known as a transcription unit. So what is this transcription unit? So I have my DNA like this, right? So this is 5', 3', 3', 5'. So we know this acts as your template strand, right? So this is your template strand, this is your non-template strand. Yes, Naveen, hello, hi. So thank you for joining my lecture. So please do like, share and subscribe. Okay, so this is my non-template and this is my template. So here in the transcription unit, I have a promoter. Promoter is the one that initiates the process of transcription. Okay, so promoter is towards the 3' end of your template strand or towards the 5' end of your non-template strand. Okay, so your promoter has to be towards the 3' end of the template strand or the 5' end of the non-template strand, and towards the end you have what is known as a terminator. Terminator is the one which stops or terminates the process of transcription. Okay, clear? Fine. So in between these two, I have what is known as my structural gene, that is actual information. So a transcription unit consists of a promoter, a terminator and a structural gene. So what is the location of your promoter? Towards the 3' end of your template strand, and your terminator is towards the 5' end of the template strand. If the positions of your promoter and terminator are reversed, then in that case you are supposed to write it with respect to the template strand. If suppose you have a promoter towards the 5' end, no, that cannot be. 5' end of the non-template strand, but if it is near this, if the promoter is near the 5' end of template strand, then the process of transcription will not happen. Okay. Yes, Munna, hello, hi. I'm good, thank you. So thank you so much for joining my lecture. So you came before Shimpi. So So thank you so much, Munna. So please do like, share and subscribe. Okay. So you understood what is a transcription unit? So transcription unit has three things. One is promoter, then you have a structural gene, then you have a terminator, right? And please make sure you write the position of the terminator and the promoter with respect to the template strand. Done? Okay, so here we talk about the transcription unit. A transcription unit in DNA is defined primarily by three regions in the DNA. Right? So one is promoter, other one is your structural gene, then you have a terminator, right? So promoter, structural gene, terminator. So here they have written your template strand on top and your non-template strand or coding strand niche. Clear? Yes? Oh, what happened to Shimpi? Shimpi is not well. So please take care of Shimpi. Okay, Munna? Yes? Fine. So please teach her whatever I am teaching. So first you understand and then you teach her. Okay, Munna? Yes? Are you able to hear me properly? Is there a problem in the sound? Yes? Is there a problem with the sound? Are you able to hear me? Yes, Munna, can you hear me? You are talking about Shimpi. Okay, Munna. Guys, can you hear me properly? Yes? Okay, yes, you can. All right. Okay, so I got the mic changed. Fine. So I hope this is clear. Okay, so now we go forward. So this is how it is. So please look into the GIF. So I had a terminator, a coding region. Now I have a promoter side. So promoter initiates the process of transcription. Termination terminates the process of transcription. Okay, done. Is it clear? Yes? So please be careful with the location of your promoter and your terminator. Okay. So now, as I told you, your RNA polymerase can catalyze only from the 5' to the 3' direction, right? So here we go. So the two strands have anti-parallel polarity. So we are talking about the two strands of DNA here. So DNA dependent RNA polymerase. So why is this RNA polymerase called as DNA dependent? Because the template is DNA, right? So that is the reason this is known as DNA dependent RNA polymerase, and it catalyzes the polymerization in only one direction. That is from the 5' to the 3' direction. But the 3' to the 5' strand on the DNA, it acts as a template strand. Okay, and this is also known as the non-coding strand, and the 5' to the 3' is known as the coding strand or the sense strand. So promoter and the terminator flank the structural gene. So this forms your transcription unit. Clear? Okay, so now fine. So the promoter is 5' towards the 5' end of the non-template strand. So this is upstream. Okay, so I told you the locations, right? Okay, so my video is stuck. Okay, I guess my video is stuck, so please hold on. Yes, I hope now it's clear. Yes, now is the video clear? Not visible. Now is it visible? Yes, I guess now I'm visible. All right, fine. So, we were talking about a transcription unit, right? So, when I spoke about a transcription unit, so here I have a promoter, here I have a terminator, and this is my structural gene. So, this is my transcription unit, right? So, promoter is towards the 3' end of the template strand, and this is the 5' end of the non-coding strand, right? So, this is the coding strand. It is given a weird name. So, this is the template strand, right? So, your RNA polymerase will bind to the promoter and then initiate the process of transcription. Once this RNA polymerase comes in contact with the terminator, it will end the process of transcription. So, this region where the promoter is present, so this is known as an upstream region. So, this is known as upstream region. Okay, the place where the terminator is present, so this is known as your downstream region. Clear? Yes? So, please remember what is upstream and downstream region. So, the same thing is written here. Okay. So, this is how it is the structure. So, before we go into the structure, so let me explain what happens here. So, first your RNA polymerase binds to your promoter, right? So, RNA polymerase, when it binds to the promoter, it recognizes a specific sequence on a promoter. So, how do I know this is Anthony Irby? Because that is how the information is stored, right? So, if I have 2 to 3 papers, if I know your handwriting well, I can find which student is that, right? So, I have some sort of recognition. So, how will your RNA polymerase recognize the promoter? So, in case of eukaryote, it is a different, and in case of prokaryotes, the recognition sequence is different. So, in case of prokaryotes, right, so you have Pranav box. So, this is T-A-A-T-A-T. Okay, so the spelling is wrong, Pranav box. Okay, so this is the sequence it will recognize in case of your prokaryotes. In case of your eukaryotes, it is the Tata box. It will recognize this sequence, TAA-TA sequence. So, your RNA polymerase will recognize these sequences in your promoter and then it will bind to the promoter. Okay? But before that, tell me, where does the process of transcription happen? So, I told you replication happens in your S phase inside your, so S phase, that is where your DNA is replicated, and this happens inside your nucleus. Okay, done. So, where does the process of transcription happen? Yes, any idea? So, where does the process of transcription happen? Yes, come on, give me the answer. Where does the process of transcription happen? Yes, quick. Where does the process of transcription happen? Yes. Transcription also happens in the nucleus only. So, no doubt. Okay. So, transcription happens in the nucleus. Translation happens in the cytoplasm. Replication and transcription happens in the nucleus. Translation happens in the cytoplasm. All right, clear? Okay, so this is how it is. Now, when my RNA, so now it is getting transcribed for me. So, RNA polymerase has bound here. So, here I have my promoter sequence. Here I have my structural gene. Here I have my terminator. So my RNA polymerase has bound and it is giving me my mRNA like this. So this is my mRNA sequence, right? Okay. Now, when this mRNA sequence comes out, it moves into the cytoplasm for translation. Okay. So, in your prokaryotes, it is different. The process of translation is different. And in your eukaryote, the process of transcription is different. How and why? So, here I am addressing your question. So, you have a term what is known as a cistron. So, what is a cistron? So, what is the gene? So, gene is any sequence on your DNA. So, gene is any sequence on your DNA that codes for a sensible product. So, this can be either proteins, this can be either RNA, rRNA or tRNA. So, proteins will produce mRNA, right? To produce a protein, it first produces mRNA. So, the gene can be any product which can code for a protein, rRNA or tRNA. When a gene specifically codes for a protein, it is known as a cistron. So, once again, so gene is such that it's a sequence on your DNA that codes for a sensible product. It can be a protein, it can be rRNA or it can be tRNA. But when it codes specifically for a protein, that is when it produces mRNA and getting translated into protein, it is known as a cistron. So, this is now not called as a gene, this is specifically called as cistron. So, in prokaryotes, we have polycistronic condition. Poly is many cistrons located together. It is clustered together in case of your prokaryotes. So, in your prokaryotes, many cistrons are clustered together. It is polycistronic in prokaryotes. So, in case of your eukaryotes, it is monocistronic. Meaning you have one cistron and then a gap and then another cistron. Okay. So, here we go how it happens. So, in case of prokaryotes, so this is my mRNA. So, my mRNA is polycistronic. So, here I have one cistron, second cistron, third cistron, fourth cistron, fifth cistron. So, this is how it is in my prokaryote. But in my eukaryote, it is different. So, this is prokaryote. So, in my eukaryote, so if this is my mRNA, right? So, I have one cistron, then another cistron. So, there is a gap between a cistron and the cistrons are not continuous. So, it is known as monocistronic. So, in my prokaryotic mRNA, I have coding sequences, the ones that code for protein. So, these are called as exons. This is exon 1, this is exon 2 and this is exon 3. So, this intertwining spaces are known as introns. This intertwining spaces are known as introns. Okay? Yes. So, this is how your prokaryotic mRNA is and this is how your this is your eukaryotic mRNA and this is your prokaryotic mRNA. So, that is why the process of transcription differs. So, now let us talk about the transcription in prokaryotes in detail. So, in case of prokaryotes, the nucleus is not well defined. So, the process of transcription, the process of translation both happen in the cytoplasm, you can say. This is for prokaryotes, alright. So, here you have to pay attention. So, your RNA polymerase has many subunits. Your RNA polymerase is a multi-enzyme complex. So it has what is known as a sigma subunit. Okay, so this sigma subunit of the RNA polymerase helps in recognizing the pronoun box or the promoter. Okay, and then your RNA polymerase produces this mRNA. And when your RNA polymerase encounters your terminator, so your RNA polymerase has another subunit known as rho subunit. So the rho subunit binds to the terminator region of the transcription unit and it brings out the RNA polymerase outside. So thus your mRNA is synthesized. Since both the process happen in the same place, as soon as your mRNA is synthesized, you have ribosomes coming and binding like this. So this is known as a polysome, if you remember. Right? So immediately just after transcription, the process of translation begins in your prokaryotes. Clear? Yes? No doubt? So this is how the process of transcription happens in your prokaryotes. So if you look at the picture here, transcription in bacteria is polycistronic. So we have three steps. Initiation, when the sigma factor of RNA polymerase binds to the promoter, then it elongates your DNA and then it terminates. Termination is with the help of a rho factor. Clear? So RNA polymerase is associated with elongation and transiently with termination. So see, promoter sequence. So I have my RNA polymerase, inside that I have a sigma factor. So this is my first process of initiation. So it will just bind to the promoter sequence. Which one? RNA polymerase, right? So once my RNA polymerase has bound, then it will lead to the elongation, that is production of my mRNA. And finally, my rho factor of the RNA polymerase will recognize the terminator and thus it will terminate the process of RNA or terminate the process of transcription. So this is the process of transcription in prokaryotes. In case of eukaryotes, it is a bit complicated just because we are multicellular organism. So here also we have three steps in the process of transcription. So first we have initiation. So in initiation, RNA polymerase will recognize the Tata box of your promoter. So this is present in the promoter region. So this is your process of initiation, right? And then you have elongation. That is, my RNA polymerase is getting produced. And then finally termination, the process of transcription will stop. Right? So the same three process. But here there is a twist. So this is my mRNA. But in my eukaryotic mRNA, if this is my exon, the next exon is here. So I have unnecessary sequence or non-coding sequences known as introns, right? So now this is not complete. So just after the process of initiation, elongation and termination, this is transcription. The RNA that comes out has both exons and introns. So such an RNA is known as primary mRNA or immature mRNA. Or it also has another name. Since this is the process of transcription, this is known as primary transcript. This is known as primary transcript. The moment this mRNA is being formed, you have post-transcriptional process. So what is this post-transcriptional process? The moment this mRNA is being formed, so the cell should ensure that this mRNA is safe and it is not destroyed. So you have some cells in your body that, some proteins in your cell that patrols and finds out anything that is unwanted, it will put it in the garbage, okay? So this mRNA has to be preserved or prevented from degrading. So the first thing that happens is capping. So this mRNA is quickly capped at the 5' end. It is capped by guanosine methyl triphosphate. So the moment this mRNA comes out after your elongation and termination process, so this mRNA is quickly capped at the 5' region. So when the degrading proteins look at this cap, they do not touch it. They do not disturb it. Okay. So this is your methyl guanosine triphosphate. So this is capping, right? So now it is now stable. So nothing will disturb it inside the nucleus. The second step, you have exons and you have introns, right? So these are not needed for us. So these have to be chopped off. So the introns will be chopped off by a process known as splicing. So all the introns will be tagged like this. So all the introns will be folded like this. Okay. So these are exons and then the introns will be chopped off. So that is by the process of splicing. Now it is ready for the process of translation, but translation happens in the cytoplasm. So now the mRNA should be such that it is ready for moving out of the nucleus. Suppose if you are going to go out, you be well dressed and it's visible from your appearance, right? So whenever some travel proteins come, so this mRNA maintains the status that it is ready for traveling. So how does it show it is ready? It has a tailing process. So at the 3' end, so now it is pretty clear, it has a 5' end and now it has a 3' end. So here it has a cap and here my exons are continuous now after the splicing process. Now to ensure and to let it know that this is very stable now and it is ready for transport outside the nucleus. So here it adds adenine residues. It nearly adds 200 adenine residues, thus it is like a tail. So this is known as a poly-A tail. Many adenine residues are added. So this is the tailing process. So in your eukaryotes, you have 3 extra steps, that is capping, splicing and tailing. Clear? Yes? Is this done? So you understood the process of transcription. Now once this tailing is visible, now the nuclear membrane will allow it to move outside the nucleus and reach the cytoplasm for translation process. Done? So gene is the functional unit of inheritance. So the DNA sequence can code for tRNA or RNA molecule, that is a gene. So then you have the definition for a cistron. So in eukaryotic it is monocistronic and in prokaryote it is polycistronic. So all these I have explained. I hope you have understood, right? Yes. So this is how it is, the primary transcript or the immature RNA. So 5', 3'. So it has introns and exons. So finally your mature RNA, your introns are chopped off, right? So here I have a 5' cap and here I have a 3' tail. So 5' cap and a 3' tail is added after the splicing process, right? So this is my mature mRNA now. Okay, so now I have something else to tell you. So this is, yeah, okay, so this is the process it happens. So I have a poly-A tail here. So these 3 process happens. Okay, so one more point that I have to mention in the process of transcription. So the mature mRNA that I have here. So this is 5', this is 3'. So now this has a poly-A tail and this has a 5' cap, right? Here I have something known as UTRs. So remember these UTRs, I will link it in translation process. Okay. So this is one term you should remember. I will tell you what are UTRs when we are talking about the process of translation. So now before that, so who catalyzes this reaction? RNA polymerase. So in your prokaryotes, you have only one RNA polymerase, so this process is easy. In your eukaryotes, you have three types of RNA polymerase. So I have RNA polymerase 1, I have RNA polymerase 2, then I have RNA polymerase 3. So RNA polymerase 1 catalyzes rRNA formation, but here our mRNA is being formed. So this catalyzes mRNA. So this is also known as heteronuclear RNA because it has a cap and a tail, right? And this catalyzes your tRNA formation. So this is how the process of transcription happens. Is this clear, Anthony and Dharini? So how the process of transcription happens in eukaryotes and prokaryotes? Yes? No doubt? No doubt? Yes? Clear? Done? Okay, so with this I stop. The process of transcription is done. So please don't forget to join me at 6 p.m. today. So we are starting a new chapter in human physiology, that is breathing and exchange of gas. So please join me for the same, and please think about joining this crash course. It has all the 3 subjects, and if you genuinely like my session, so please do like, share and subscribe. And thank you so much for joining my lecture. So this is me wishing you more and most, waiting to get connected with you, and yes, I'm ready to help you in any manner if you have doubts in biology. So please feel free to ask me. Thank you so much. I'm looking forward to seeing you at 6 p.m. So until then, stay tuned and take care. Bye.